So good afternoon and welcome to the CFVGA, the Colorado Fruit and Vegetable Growers Association Lunch and Learn webinar, Salmonella in the Onion Packinghouse. I'm Adrian Card, State Specialist in Produce with Colorado State University Extension and founding board member of the Colorado Fruit and Vegetable Growers Association. As your host today, this webinar is produced in collaboration with CFVGA and will explore where we can find Salmonella and other human pathogens inside a dry bulb onion packing house based on equipment materials, sanitation practices, and onion scale. CFVGA is the go-to resource for Colorado's fresh fruit and vegetable farmers. We connect Colorado growers with industry, government, academia, and consumers to strengthen and expand Colorado fresh fruit and vegetable production. Learn more at CFVGA's website and social media channels. CFVGA is made possible by the generous sponsorship of organizations in Colorado, and the board would like to thank its sponsors today. Here are a few of the platinum sponsors on screen. Gold sponsors, silver sponsors, and finally, the remaining silver sponsors. Learn more about these businesses on the CFVGA website on the Grower tab and explore their products and services. We'll do Q&A at the end of Eduardo's presentation today. Feel free to type those questions into the Q&A box as we go. You can see all of the questions that everyone's typed in, so make sure you reference that. You can kind of see what's emerging as thought as we go. And we'll have an educational poll at the end of today's webinar. Our presenter today is Dr. Eduardo Gutierrez Rodriguez. He is an assistant professor in food safety systems in the Department of Horticulture and Landscape Architecture at Colorado State University. Dr. Gutierrez Rodriguez has over 20 years of research, extension, and business experience in food safety, focusing on fresh produce and minimally processed food environments. His research centers on understanding the microbial ecology of human pathogens within farm and food processing environments, looking at microbial interactions, risk assessment, and remediation practices that can sustain fruit and vegetable production and reduce transfer of human pathogens into the food supply chain. And with that, Eduardo, we'll pass over to you.
Thank you, Adrian. Let's grab the controls of the spaceship here. And then we will turn these on. Okay. Alrighty, guys and ladies. Thank you very much for being here with me today. Last week, we talked a little bit about actually contamination of Salmonella in the field. And today we're going to basically focus on contamination of Salmonella in the packing house. Again, just want to make sure that I acknowledge those people that have done the majority of the work. The Specialty Crops Block Grant Initiative, HR 133, that provided funding.
Eduardo, is your video on?
It's my camera? Video. Okay, it should be on, but let's see.
Sorry to interrupt you.
No worries.
Didn't see you there for a second.
Okay, are we good?
Yeah, and just back to screen share.
Okay, screen share, sorry.
Yeah.
Alrighty.
Brilliant.
All right, so my apologies for that. So again, we're talking about the packing house today and just making sure that I acknowledge Susan and Jeffrey from Fresno State, Tony, Griffin, and Mark from Colorado State that helped us quite a bit, especially Griffin as a graduate student, Marilyn Drake, and folks from Colorado Fruit and Vegetable Growers Association and the Onion Group that has supported us quite a bit, Joe Petrocco and Sakata Farms for their guidance around this, Jebima and Kevin Tanabe that help us with onion production back there in La Junta. And then, of course, folks here from Research Station, Sky Handyside, and Hannah Sutherland, and Colorado State, and the graduate students that worked in the project. Today, like I said, we're going to focus on the packing house aspect. So for those of you that were here, these were some of the pictures that we showed last week, in which on the top you see the onions packed and ready to move or transport from Colorado to California. And on the bottom, all the onions now in the spaceship ready to cross the mountains and get over to Fresno, where we were going to do all our packing activities. So what I'm going to quickly do is just a quick reminder of our field results. Then we're going to get into the risks during harvest and storage of these onions, and then into dry sanitation and the key findings. So just to make sure, we're going to focus here on packing house activities. And so the concerns for packing house activities were mainly three. One, sanitation-wise, can there be cross-contamination going on? How does that dry sanitation or wet sanitation work? What is the risk of contamination between onions to the packing line or to the onion? And whether that contamination would basically transfer over time to the storage conditions or to the bins. That was the basic premise behind that. So what we were trying to do, again, is to try to do a comprehensive assessment of harvesting and storage practices and how variety, season, and post-harvest handling were going to attribute pathogen survival. That's been the goal. So when we talked about production practices, the summary from last presentation was that onion cultivar did not impact the survival and persistence of Salmonella, that Salmonella was able to survive across basically the whole entire cropping cycle. The closer you got to topping and lifting, the higher the risk and the higher the population of Salmonella. And some of the other things that we didn't share with you last time that is important to mention here today is that we looked at soil moisture content and whether that had a positive or negative impact in the survival on Salmonella, and we didn't find that. And then when we looked at UV index, we found this dichotomy that it does help in the reduction of Salmonella in the onion, but it doesn't do anything in the soil. So if the soil is contaminated, even if we have a high or low UV index, it just basically stays there. But we do see some differences in the survival of the onion. We're not exactly sure if it's a really direct correlation with UV index or if it's more an issue of stress of the Salmonella from being in the field. But irrespective of that, what I just want to remind you all, and where we're going to start today, is here where it says harvest. And so we had onions that were at the 2 to 3 leaf stage, which we had a population of about 10 cells from the 2 to 3 leaf stage, meaning the contamination happened early in the season. And then we had 2 other contamination events that happened during topping and lifting, in which at the moment of harvest, we had about 1,000 cells on each of them, 1,000 cells of Salmonella on the onions at the moment of harvest, okay? So those were harvested, and when they were harvested, they were put in these bins, and these bins were transported to California. So when they arrived in California in these bins, we understood that the population was going to be kind of like that. We had a high contamination event in which we purposely contaminated them with chalk so that they would have a very big population. We had the population at topping that was about 1,000 cells, and then we had the population from the 3 leaf stage that was between 10 cells or so. And just to make sure that we're all speaking the same language, this is how we would see this. We would have a high risk, like I told you, about 100,000 cells, in which, if you can see the picture, the yellow onions have a little bit of a white powder on them. That is that chalk that had the Salmonella. Then we had these medium risk onions that had about 1,000 cells of Salmonella at harvest that came from the topping and the lifting. And then we had the low risk Salmonella that, or onions that had about 10 cells at the moment that we harvest them. And so the obvious question was, what would happen once we harvest them and we transport them and store them in California before we do the sorting and packing? So this is what happened. Basically, within a span of 15 days, whatever was considered high risk, medium risk, or low risk, basically all the Salmonella disappeared within 15 days. And it went below 10 cells of each of the treatments. Of course, at the time that we were doing the evaluation, we had no idea that this reduction was going on. And as we began doing the analysis, once we arrived in Fresno and started collecting the samples, then we realized that even those cells, those onions that had 1,000 cells, 100,000 cells or 1,000 cells, had basically gone down all the way to 10 cells. So basically what happened then is at the moment that we started doing all the swabbing and all the processing in the facility, most of the onions had only 10 cells or so that started going through the packing line. So to succinctly summarize this, we saw a significant reduction between harvest, storage, transport, and storage. But the interesting part is that the population stayed the same irrespective of whether you had a high contamination, a medium contamination, or a low contamination. What does it mean? It means that there is a small amount of this Salmonella that will survive and that is going to persist along the different steps in the process. The good news is that there was a big reduction, more than what we expected, but still it was there at the moment of processing. So let's talk about the processing and packing house activities that we were talking about. So here, again, on the left side, you see the bins when they arrive from Colorado. And when you, on the right side, then you see an example of where the onion process, sorting process started, okay? Just to make sure that we all are in the same understanding, one of the things that is important in any packing house is being able to understand the zones. So we have zone 1, which is direct contact with food. Zone 2, which is adjacent, very close to that food contact surface. Zone 3 could be the drains, or it could be the legs of the table. And then zone 4 would be something outside of the facility. We try to focus in this evaluation only in zone 1s, because those are the ones that are going to be food contact surfaces. One of the other things that is important to mention is that in packing houses, we have all these different types of metals. We can have stainless steel, we can have plastic, we can have other types of brushes and bristles that may be made of plastics or camel hair or other things. And of course, the sanitary design of all of them is going to impact our ability to recover that organism. Specifically, for example, if you have welding like the one that you see before at the bottom of the screen, or when you have some sort of chafing or some sort of indentations in the materials. All of these were present in the packing line that we were doing all our evaluations, and typically represents what you would see in other packing house events. And so the question is, are these materials impacting the survival of Salmonella? Are these materials impacting the survival of other indicator organisms? And the question is are these materials cleanable, right? Because at the end, we want to make sure that we have as clean as possible as a facility. Why is that a problem? Because all these onion residues, the scale of the onion, if there is issues in the quality of the onion, it leaves these residues in the line that are the perfect harborage for biofilms. And then once here's a cartoon of how the kind of the biofilm is being created. You have a little bit of attachment at the beginning that happens in between some of those residues. The microbial population begins to grow. And at some point when it's big enough, then basically what it's doing is just as soon as the onion roll over, it grabs a little bit of that biofilm, takes it with it, and continues going. So depending on the material and depending on the accumulation, we may or may not have more biofilms, okay? So how did the packing line look like? So here are pictures that I want to show you. So for example, at the very beginning, here we had these plastic PVC rolls. And if you see here, somebody was doing a dry sanitation event here with alcohol wipes, and we're going to talk about it a little bit more. So that was one of them. Then in this number 3 here, you see a stainless steel trough that basically all the onions land into before they start going into the brushes. Here you see some of these plastic brushes. Here you have the typical curtains on number 6, another metal trough. And here in number 5, you see another type of brush that is used or was used in this packing line. And then as we continue on into the different sections of the packing line, you can see at number 8, a conveyor belt, that there's plenty of plastics and conveyor belts in number 8, or on number 9, that you have these wedges of plastic in which the onions work. And so if you see someone here, they had a single-use paper towel and food grade alcohol that we were hoping to use for dry sanitation. And then on number 10, it's where the onions basically fall and begin to be going to separate based on USDA grade, basically the size, the weight, and so on and so forth, their caliber. And then on 11 and 14 is these locations in which the onions landed that are all stainless steel, in which the onions were separated by USDA grade. So all these surfaces we swabbed because we wanted to know, basically, is the onion leaving residues, which most likely it is Salmonella staying in those residues? And what about the indicator organisms? Are they telling us anything? And if we know those 3 things, then the obvious question is can we do a dry sanitation event? Why a dry sanitation event? Because onions don't like water. We don't want to have water in onions. If we have water in onions, then we're going to have issues with plant pathogens and deterioration of the crop. So we need to make sure that we apply some sort of sanitation procedure that allows us to reduce that residues, that cross-contamination that can happen. And so we're going to get a little bit more in detail about that dry sanitation aspect. Before we get there, I just wanted to make sure that we were all in the same page about how do we go about testing surfaces, okay? So these next 3 or 4 slides talk specifically about how we go about testing of surfaces. So we can use ATP, like you see here, and basically ATP, what it's measuring is the residues of the food, the scale, the liquids, anything that is organic matter related, all those debris is what we measure with ATP. We can also do swabs, like you see here with this white sponge here, in which we actually are swabbing the surface, not for ATP, but to be able to get a microorganism out of a surface. And when we do these things, the pressure that you use to swab is critical to be able to remove those organisms. And the last thing that is critical in this testing approach is making sure that you use the right microbiological technique, because many of the cells that are in those packing lines or in those situations are stressed. So they're not in perfect condition. So they could still be viable, but not culturable. So we need to have the right microbiological techniques to be able to culture them, because if not, we're going to get a lot of false negatives, even though they're still there. So the message here is ATP is only for debris. If you're testing for human pathogens or for indicator organisms, make sure that you use the right swab and make sure that you use the right microbiological technique so that you don't lie to yourself. Why is it important? Because we can have these microorganisms. In this study, we wanted to work with Enterobacteriaceae and E. coli, which are the typical indicator organisms that are used when swabbing surfaces. And those are typically used to potentially, and I want to put in quotation marks, potentially describe the presence of Salmonella, pathogenic E. coli, in this case, E. coli 157, and in some instances, Listeria monocytogenes. The focus of this project was on Salmonella. So our goal was to see whether Enterobacteriaceae here, EB, or E. coli, were going to be good predictors of the presence of Salmonella in the packing line. This is the message that I want you all to hear. Thus the indicator organisms, which one? E. coli and Enterobacteriaceae, are good predictors of the presence of Salmonella. That's what we wanted to do. And we use the techniques that I showed you before, ATP for debris and the swabs for determining the bacteria. When we're using ATP, there's different brands. There are many different brands there. And some of these brands have different parameters, okay? The brand that we use basically uses a parameter that any value of 30 or below is considered clean. There are other companies that use different thresholds. But one of the things that we enjoyed and liked or appreciated about the materials that we were using here is that this company has a positive and a negative control and a calibration system. Why is that important? Because every time that you do the measurement, you can calibrate the equipment, you can do a positive test to make sure that you are in the range, and then you make the measurement. So in that way, you're always guaranteeing yourself that every time that you do a measurement, the measurement is consistent within the parameters of the equipment, and also allows you to verify whether your measurements are being done accurately. So in this case, what we measure is the relative light units, and this RLU that is here at the bottom of your screen, and that relative light units is reflective of the amount of debris or ATP that is in the surface. And what does it mean? If you have a very low RLU, if you have a very low value, it means that most likely it's clean. And if you see here in this graph here on the right, the sharper the arrow goes, or the higher the arrow goes, the dirtier the system is. That's just the gist of how it works. So what we're hoping to see is when we do the swabs with ATP, that we're going to have a very small value so that we know that we're clean. Okay, and we want to evaluate whether the dry sanitation does work, and we use this to determine that aspect. So again, if you're going to venture into ATP use, make sure that you know the brand, Make sure that you understand that this is only for residues of organic matter. Make sure that your material or the equipment has a calibration system, has a positive and negative control, so that you can be confident of those values. And that's basically what we use in this system, okay? And the last thing that is important, not only for ATP, but also for swabbing, is the way that you do the swabbing. So in this picture, there is a description that it goes 1, 2, 3, and 4. So basically, that is the process that we used to follow. 1, then opposite 2, then opposite 3, and then opposite 4. And in that way, you cover that specific area. And one of the things that I want to make sure that is clear, if you look at the bottom here, it says correct and wrong. Why is that? Because the one that says wrong is just no pressure. There's very little pressure on the surface. You really need to pressure down that swab to be able to remove these residues for ATP and also for bacteria. So some of the employees tend to be afraid that they're going to break it. If they break it, awesome, grab a new one. We want these guys to be basically pressuring down the swab as much as they can, with, of course, without breaking them, so that they can remove some of those residues from it, okay? So just want to make sure that we have that. So when we were doing our swabbing evaluations, everybody that swabbed was trained in this matter to make sure that they were consistently grabbing the bacteria or anything that was left there in the same manner, okay? So now that we've sort of done this initial discussion of how we went about things, let's go back again into the results of the packing line, okay? So the swabbing method is this one. This is the ATP luminometer that we used. Here is an example of the area that we were swabbing and one of the measurements. So we established a baseline of the line, and then from there we did dry sanitation events, and then we passed the onions to see how everything was going on. The dry sanitation approach that we followed is this one. We started first with the scrubbing with single-use paper towel and a little brush. We followed this by an application of 70% food-grade ethanol. That 70% food-grade ethanol was then removed or scrubbed away with single-use paper towel. And then we finished up with food-grade alcohol wipes that basically you go in the same area, wipe the area, and then you let it air dry. Why? No water residues. And in that process, you are scrubbing and using sanitizers that may allow you to potentially reduce either indicator organisms or the human pathogen in question, okay? That was the sanitation approach that we followed. And we had 14 different locations that we were going to swab consistently every time to make sure that we were consistent in the measurements. So here's the first slide. I know that this is a busy slide, but what I want you to understand is the following. We're gonna go to this panel here on the left that it says stainless steel leaf eliminator. Okay, this first bar here that it says DS1, this is when we did the dry sanitation, okay? So the value that we got after dry sanitation was 70. We were not there yet. We needed to get to 30, but we were close. This second bar here, the one with a B here, it's when we started passing the onions during the entire day. So we grabbed the onions with the high risk and we started selecting the onions during 4 hours. So after 4 hours, this is the amount of onion debris that were left in the packing line, okay? We didn't do any clean sanitation because this is typically what happens in activities in the onion line. So the next day, we came with the yellow onions, with the next set of onions, and we passed them again during 4 hours. What happened? The amount of residues started increasing more and more and more exponentially, okay? So after 2 days of running onions with no sanitation, what happened? The accumulation of debris was significant. So if you see this square here, it is the same approach here. It is the same approach down here. It is the same approach here. It is the same approach here. And basically, in different plastics, in different stainless steels, most of the time, the accumulation was the same. The brushes was the nightmare because we couldn't clean anything. It was dirty all the time. But in any other surface, if there was not a sanitation event between one event and the other, the accumulation went up. So if we go back again to this stainless steel leaf eliminator here, after the second day of processing, then we did a dry sanitation. And what happened? We almost hit the target. We had a 34. So we went from almost 4,000 units of ATP, which means filthy, beyond filthy, to be colloquial, to almost passing the requirement of being clean. Okay? So what is, and this was from high-risk onions. So what's the message here? If you do not clean and sanitize after every day of operation, the amount of debris will continue to increase exponentially. And at some point, you're going to have the conditions necessary for a human pathogen to grow and survive. So the dry sanitation approach that we're following here seems to be effective. This was with high-risk onions. Why the high-risk onions? Those were the onions that supposedly had 1,000 cells, 100,000 cells. Then, when we did this with the low-risk or medium-risk onions, the ones that had 1,000 or less cells, we pretty much saw the same thing. When we do the cleaning, we have a 34 here. If we stay again in the leaf eliminator, okay, we have a 34, so we're almost clean. Then the first day happened, the accumulation went up again. Then the second day happened, and the accumulation kind of stayed the same. Even though the bars are small, they were not significantly different. But as you can see, it stays the same, it stays the same, stays the same, stays the same, it stays the same in all these. What are the two key messages here? These are low-risk onions. These onions have less scale, had less contamination, has less things. So if you can see the ATP amounts that we're measuring, it's in the 2,000 range. Okay, if I go back to the previous one, we were in the 4,000 or 5,000 range. So these ones had a lot of debris, a lot of residues, a lot of scale. These ones had much less. So of course the accumulation is smaller. But again, we see the same trend, and we see the sanitation, dry sanitation event is able to reduce the population. If you see on the other stainless steels, we went from 2,000 to 108. In this one, we went to 2,000 to 114. In this one, we didn't do a great of a job. We went from 1,500 to 446. But still, we are able to reduce the population, or we're able to reduce the residues, which is important, okay? So now let's move to what is the indicator organisms. So this is also a busy slide about indicator organisms and what happens with them in the packing line. So the black bars are Enterobacteriaceae, and the gray bars are E. coli. So in this case, as you see here in red, the dry sanitation was marginally effective. If you see here, for example, on the stainless steel leaf eliminator, this one that has a C is when we did the sanitation. So we still had some Enterobacteriaceae, but we didn't have E. coli. The next day, the population of EB went up. Still, we didn't have E. coli. The third day, the population of EB went even higher, and now we find E. coli. How does that correlate to what I said before? We start building the amount of debris, and at the same time, we're building the population of indicator organisms, of E. coli and Enterobacteriaceae. So the dry sanitation event was able to reduce those populations, not enough, was able to show improvement in between runs, and was able, if you see here, able to eliminate, in this case of this stainless steel, all the EB and all the E. coli. But it didn't happen in the brushes, which I told you that the brushes were basically impossible to clean. But in most stainless steel, we were able to do it. If you see this vinyl plastic here, we had success with E. coli. We eliminated E. coli, which is great, but we were still able to leave some of Enterobacteriaceae. And if we go to this other part here at the bottom, it's the same thing. We reduce the population of EB and we eliminate E. coli. So it's being good at reducing these organisms too, besides the residues. And we see the same effect when we're talking about low-risk onions here. And again, those that were in very low populations. Okay, the only difference between these ones and the other ones is that the population stays much more stable, much more stable. Okay, so what is the message that we want to convey to you guys? There are differences in the accumulation of debris and in the accumulation of bacteria depending on the material. In this example here, if you see, these are only surfaces that have stainless steel. And we're talking about sections 11 and 14, section 7, section 3, and section 1. Only those between high risk onions that had a lot of potential salmonella and low risk onions that had little salmonella. So the accumulation or the trend or the rate of accumulation is always going up, but the rate of accumulation is different. In this one, it's a very sharp accumulation rate because there is a lot of debris, a lot of residues. In this other one that has less residues, the rate of accumulation of the bacteria is much flatter, which is what we were talking before. Okay, so what does it mean? That if I don't have a sanitation event, the accumulation is going to continue, but the accumulation is going to change if you have a lot of debris versus you have less debris. And we see the same thing here with rubber belts. We did this for rubber belts, and again, we see the high and low risk, and what is the trend? One very sharp versus another one that the trend is much lower. Okay, so what is the message so that we can open this up for questions? Salmonella is capable of surviving and persisting during transport and storage. We did see salmonella still there, but what we saw was a big decline. Dry sanitation worked. Dry sanitation was able to reduce residues of dry bulb onions and was able to reduce a little bit of Enterobacteriaceae and E. coli. But the interesting part here was that there was no correlation between the presence of Enterobacteriaceae and E. coli and salmonella. In all this testing that we did, we never recovered salmonella from the surface of the onion or from the surface of the facility. Okay, why? Because when we started the population or when we started the experiment, remember, we went from 100,000 cells to less than 10 because it died during storage and transport. So we were not able to recover it in the packing line. Why? Because it's a huge packing line and because it's being diluted because it only has 10 cells. But what is the message? Salmonella is going to find itself in one of those niches that we don't clean or in areas that have debris. And if we don't clean those debris, then those debris are going to accumulate and accumulate and accumulate up to a point in which then what happens? The conditions are ready for salmonella to grow and to cross-contaminate. Okay? So what is the message that we want to share with you? We think that you have to include a daily dry sanitation event in your packing line so that you are consistently being able to reduce the residues and are consistently able to reduce the indicator organisms. We don't think there is a correlation with, or at least the data that we have here, doesn't suggest that there is a direct correlation between the presence of an indicator organism and a human pathogen. And that's one thing that we need to explore more to find a definitive answer. But I think that ATP measurements are going to give you a very good understanding of the level of risk that you have. If you have high ATP, you have high risk. If you have very little ATP, the risk is lower. However, that doesn't mean that there could not be a pathogen, right? What it means from the standpoint of risk is that we have less food available, less of these things that the organism mean. It doesn't mean that we don't have a human pathogen, okay? To be able to know if we have a human pathogen, we have to be able to test for that human pathogen. But if we don't clean or we don't have these dry sanitation events, the risk increases and increases and increases daily. And with that, I want to basically stop and open this up for questions.
Thank you, Eduardo. We're getting questions here in the Q&A. Do you want me to read these aloud?
Yes, please. That might help. Great.
Yeah, that might help our people watching this recording. I wonder, JB asks, I wonder where the 3 steps of testing, 5 days after harvest, 5 days after transporting, 5 days after storage, were they separated tests or consecutive tests? There are 3 parts to this, maybe 4. Maybe we stop there. Yeah.
Sure. No, JB, when we harvest the onions, we did the test. When we inoculated the onions with chalk, we did the test to know what is the initial population. Then we put them in the boxes. Then we put them in the truck, and then the truck drove. When they arrived to California, we stored them there. And the day before that we started doing the analysis, we collected the onions to see what was the population. So during those 13 to 15 days, the salmonella died exponentially as while they were storing.
Mm-hmm. Why did you choose the 5 days as a timeline?
We only choose the 5 days of the timeline because of the transport aspect of things. I didn't mention that we also had a separate study in which we stored the onions for 3 months in burlap sacks at the typical relative humidity and temperature that most commercial operations do. Those also came from the high risk, the medium risk, and the low risk. And when we did that 3-month study, after 3 months, we were not able to detect Salmonella in any of the onions. We stored 32 burlap sacks with about 20 kilograms each for those 3 months. And when we went back to do the test, we did most probable number, which is a technique that allows you to detect almost very little populations. We did enrichments, we did genetic testing, and we were not able to recover the Salmonella from any of them, from the high risk, from the medium risk, and the low risk. So during those 3 months, of course, there was some significant die-off that was happening. We cannot guarantee for sure that Salmonella was not there, but at least we were not able to detect it, even though under those extreme inoculation conditions.
Great. And JB also asked if some packing houses use water to wash out field soil. How would that impact this Salmonella concern?
I personally haven't seen an onion facility use water to remove some of the soil from the onions, so maybe I'm misunderstanding. I know that when the season begins, they do a thorough cleaning of the whole unit, and then as the sorting and processing begins, depending on the state that you are, some may have some sanitation events, some may don't have any sanitation events. But my experience in general is that there is no water in any packing line associated with dry bulb onions. So knowing that water leads to plant pathogens and decay, we wanted to develop a dry sanitation system that would allow us to eliminate, like I said, residues and as many indicator organisms as we could, so that then the risk of contamination would be lower from two perspectives, from the human pathogen side of things, but also to not increase plant pathogen incidence if they were sorted and stored.
Great. Well, this is a two-part series. If folks are watching this now, the first one was a Salmonella in the soil, how to prevent that. And you led off with, this is in direct response to the Salmonella outbreaks in 2020 and 21. And it seems like your research, as I'm listening in on this, we're still uncertain how Salmonella is spreading. Where do you go next with this to try to decipher how do we control for this and prevent it from happening in the future?
I don't know, maybe I'm weird, but I tend to think about this differently.
Yeah.
There will be a moment in which our farms may be exposed to a human pathogen, and we may know it, we may not know it. What I think we need to do as a grower is put in place the hurdles in the system that would allow me to catch that potential situation that happens. So if I have a packing house that I never clean and sanitize during 3 months or during a month or during 2 months or whatever time it is, I think we have enough evidence here and very solid evidence to say that if you apply a dry sanitation event, you're going to reduce the risk quite a lot. Okay, so do you have it or you don't have it? Did the chicken or the egg came first? Whatever. It's like, what am I doing in the packing house to be able to reduce that? And from the other study in the field, we know that if the contamination happens at the beginning of the season, it's going to stay there. And if it happens late in the season, it's going to basically stay even more and higher. So what I need to do in the farm is put a hurdle to reduce the likelihood of contamination. In onions, it's kind of hard because why? If you have furrow irrigation, you can't treat the water. If you have drip irrigation, then you could. If you're going to do sprinkle irrigation, then you could. So if you had furrow irrigation, then your risk is going to be much higher because you cannot treat that water. So if you could switch to furrow, from furrow to drip, or from furrow to sprinkler, in which you have the ability to treat the water, then you're reducing the risk there. And then if you think of land history, what is the rotation of the crop? What was it there before? What kind of situations were there before? That gives me an idea of what level of risk do I have. And so from the outbreak, it looks like the contamination happened because there was a lot of heat, there were a lot of animal operations, and sanitation was iffy in the packing line. With what I told you before is, okay, if I have a good sanitation event, maybe that takes care of it. Oh, if I have a dry spell and I have a problem with the onion, well, then it's a decision. Do I harvest? Do I don't harvest? What are the things? And that's the hard part that I can't answer, right? Because we're talking about millions of dollars that a grower has to decide whether to harvest or not, right? But if you did the history, I'm close to an animal operation. Ooh, the risk is pretty high because I'm close to an animal operation. The water system may be contaminated. Am I doing further irrigation? High risk. If I'm doing drip, maybe not. So I unfortunately, or fortunately, I don't know how you guys see this, but I think at the end it's all going to be based on how do I evaluate the risks that I have in the practices and what hurdles can I put in the system to reduce it as much as I can.
So really back to sort of that produce safety rule framework of risk reduction, what we can do to improve practices like you described there, the irrigation component, which if you're reading in the Q&A, JB's pointing out a farm in an Imperial Valley that does do the water, and that as a pre-wash in onions, and that seems like that could be, if you had Salmonella present, could spread it around more.
Yeah, pre-wash, yep. I think that every person has a different way of peeling the onion, and so I'm really not gonna sort of get into right or wrong or this or that. I think that the important part here is that we know that dry sanitation or dry conditions are not supportive of the survival of most human pathogens. The drier you are, the potentially safer you are. But we know that Salmonella and Listeria can survive in those dry sanitation situations, in those dry conditions, if there is no good sanitation. So we need to try to figure out a way to introduce into our dry packing lines, dry sanitation activities that can reduce that level of contamination. Because Listeria is an environmental pathogen. It's going to survive. We saw it in the caramel apples. It stayed 7 months alive in a dry apple packing house, and they were able to pick it up. What happened during those 7 months? Nobody cleaned it.
Well, Eduardo, it's been really illuminating. I don't see any other questions in the Q&A. So with that, I want to thank you so much for being with us today and sharing your research.
No, thank you for having me, and hopefully it will help in some way, and happy to contact or talk to anybody that if we want to talk about it more in detail.
Sounds great. Thanks, everybody. Have a great day.
Okay, bye.